So now that we have talked about how we go from a gene to a protein, we are going to talk about how these genes are regulated. So you can imagine not all genes are needed to be expressed all the time, or not all genes are going to be expressed in every cell. So looking at, for example, a liver cell. So livers have a function, many functions, but one of them is to detoxify toxins from your body. But you have the same DNA in all of your cells except your gametes. But you have the same DNA in all of your cells. So you have the same DNA in your heart cells as you have in your liver cells, as you have in your skin cells. But they have different functions. So a lot of genes that are turned on in one cell are actually turned off in other cells. So like the liver, the genes that would be turned on in a skin cell, for example, would be turned off in a liver cell, most of them. So not all genes need to be expressed all of the time. There are genes in different cells that never get turned on. So another example is the eye cells aren't going to have the liver function genes turned on. And genes are turned on and off as needed. So we're going to look at introductory how genes are regulated, but if you go into university and you study genetics, there's a whole field of just how genes are regulated, depending on the gene in the cell in the situation. So housekeeping genes are genes that are always expressed as they're always needed for the functioning of a cell. So you can think of what all cells need to do. So all cells have cell membrane, proteins, transporters, vesicles. All cells need energy, so there's going to be a lot of metabolic proteins that are turned on in all cells. All cells are going to need DNA transcription and translation. So all of those are considered housekeeping genes, genes that are needed no matter what type of cell or essentially what type of conditions that the organism is in. So we're going to talk about two scenarios and their opposite situations. So the lac operon is extremely popular in science. I use this in my master's to figure out what transporters that Sunrhizobium mellilodi had. So we're not going to talk about how this is used in my research, but it's very popular, and its role in science is very important. But we're just going to talk about how it works. So not really how we use this to study things, but just its role in bacteria itself. So lactose is an energy source for prokaryotes, and it has to be acquired from the environment. So bacteria can't make their own lactose, so they're going to get it from the environment. So let's look at what a typical, most operons will look like this. And an operon is a set of genes that are downstream from one another, and they all work together to accomplish a similar task. And they're all going to look similar but different. So starting here, we have our promoter. So this is shown in green. So this is essentially where RNA polymerase is going to bind and start transcription. We have this operator right here, which we're going to talk about. So this is a special sequence where something like a repressor, for example, would bind to this operator. And this repressor is going to stop any kind of transcription from happening. So if there is this repressor sitting on this special sequence called an operator, then the RNA polymerase cannot bind. So that's where the stop sign is. Then we see the stop sign again saying transcription is blocked. So when this repressor is attached to the operator, then the RNA polymerase can't bind to the promoter and transcribe the set of genes. Now, let's just look at the genes that would be transcribed if they could. So we have three genes. So we have LAC-Z, LAC-Y, and LAC-A. So these three genes would be transcribed to produce three different proteins. all involved in the metabolism of lactose. So this would do one job, this protein would do another job, and this protein would do another job, all to turn lactose into something usable for the cell. So here's some wordage for the definitions. So the promoter is where DNA transcription begins. So again, this is where the RNA polymerase would bind, unwind, and start transcribing the pre-mRNA. The operator is a sequence of DNA following the promoter. This is the region that regulatory factors, like a repressor, would bind to, and this controls gene expression. So as I mentioned, in this scenario, when this repressor is bound to the operator, then transcription can't happen. So it's a way to control whether these three genes get transcribed and then translated to their proteins. We also have a repressor protein, as I mentioned here. So this is the actual protein, but here is the gene. So this is a LAC-I gene. This codes for the repressor. So when this gets transcribed to an mRNA and translated to the protein, a LAC repressor, then it will go and bind to the operator and prevent transcription of these downstream genes. So we're going to talk about two situations. The first one we're going to talk about is when lactose is absent. So lactose is always potentially metabolized by every cell. And we're talking about prokaryotic cells again. So the cell is going to take cues from the environment, from the concentration of lactose. So is lactose absent, then this is the scenario that is going to happen. So the gene for the repressor, so the LAC-I gene, is always transcribed. So it's always being turned on. So this repressor is always being produced. Okay, it's important for you to remember that. So this is always being produced, so it needs to be regulated. Otherwise, it's just always going to be stopping this transcription, and then we're not going to get metabolism of lactose when it is present. And again, remember, the bacteria need lactose as a source of energy. So this is always being produced, so the LAC repressor is always present. So when lactose is absent, this repressor is going to be present, it's going to bind to the operator, and it's going to prevent transcription of these genes. That makes sense, because if you don't have lactose present, you don't need to metabolize lactose, so these don't need to be made. It would just be a complete waste of the cell's energy and resources to make proteins that are going to metabolize something that's not there. Okay, so that's why this repressor would be present and would be stopping transcription or preventing transcription from happening when there is no lactose present, because there's no lactose present, there's no need to be trying to metabolize it because it's not there. However, when lactose is present, it's a great energy source, so it needs to be metabolized. So what happens is this still is always being created, this LAC repressor is always created, but if there's lactose present, this lactose itself is going to bind to the repressor and deactivate it. So it's considered an inactive repressor when lactose is bound to that repressor. When it's bound to the repressor can no longer bind to the operator. And then when the operator doesn't have the repressor attached to it, then we can have the RNA polymerase bind to the promoter and transcribe those genes. Once those genes are transcribed, they get translated, and we get the 3 proteins needed to metabolize lactose. Isn't that brilliant? It's brilliant. So let's read through this, just to make sure I haven't missed anything. So lactose is going to bind to the LAC repressor to inactivate it. So this repressor cannot bind to the operator when it has lactose bound to it. Transcription of the lactose metabolizing genes can then occur. The lactose metabolizing proteins can then metabolize lactose. Now, what's brilliant is as the lactose gets metabolized, your lactose concentrations decrease. That means there's going to be less lactose to deactivate the repressor, and then the repressor will then be able to bind to the operator and stop transcription because you're running out of lactose. So it's a brilliant cycle of how the lactose will actually regulate its own metabolism. So when lactose is present, it stops the repressor from stopping transcription of the genes that will ultimately metabolize lactose. Okay, so lactose is the signal molecule. Lactose is what's in the environment telling the cell what to do. It is considered an inducer. It's a signal molecule that triggers the expression of an operon's genes. So with the presence of lactose, the lactose metabolizing genes can be expressed. It's considered, the lac operon is considered an inducible operon. So the presence of lactose, the inducer, inactivates the repressor and allows, again, transcription to happen. There is a direct correlation between the amount of lactose in a cell and the rate at which the lac enzymes are synthesized. So the more lactose present, the more repressors will be inactivated, the more transcription can occur. Okay, so let's look at an opposite situation. We have the trip operon or tryptophan operon. So hopefully tryptophan, we remember the sound of it. It's an amino acid that prokaryotes can synthesize, so they make it themselves, or they can take it up from the environment. It's easier for an organism to take it in than to make it themselves. It's not a hymn. And it looks a lot similar to the lac operon, where we have the promoter upstream, we have an operator, and we have 5 genes. So instead of 3, there's 5. Trip E, trip D, trip C, trip B, and trip A. So there is a trip repressor protein, again, which is always expressed, so it has to be regulated. So upstream, we have this trip R, and this, again, is going to be transcribed and then translated to the trip repressor protein. But this is opposite. So this trip repressor is inactive. Now let's pause and talk about why. So this operon is not going to be transcribed and translated to break down tryptophan. These are actual genes that are going to work together to produce tryptophan. So it's the opposite situation. So the trip repressor protein is activated by the presence of tryptophan. So pause. If tryptophan is present in the environment, then the cell doesn't need to be making tryptophan. So the presence of tryptophan is going to make it so that this becomes active and then will stop this operon from being transcribed. Because if tryptophan is present in the environment, the cell doesn't need to make its own tryptophan. Okay, so similar but different. So it's backwards thinking from the lac operon. So let's talk about the two scenarios. So again, remember, we have the trip R always being transcribed and translated, but this is an inactive form. So this isn't binding to the operator. So when tryptophan is not present, this is inactive. And we can have RNA polymerase bind to the promoter and transcribe the 5 genes needed to make tryptophan. So tryptophan is being made by these genes, ultimately being transcribed and translated. So these are going to lead to the production of tryptophan by the cell. However, if there's tryptophan present in the environment, or if the cell has a lot of tryptophan floating around that's not being used, then tryptophan would actually bind to this regulator and activate it. It will then bind to the operator and cause transcription to pause because there's so much tryptophan in the environment or just in the cell that has produced too much. That's going to pause the transcription. We don't need any more tryptophan. Stop it. Stop making it. And that's what happens when we have tryptophan present. Okay, so tryptophan is considered a co-repressor. So this is a signal molecule that binds to the regulatory protein to reduce. So co-repressor, it's repressing. So it's going to reduce the expression of the operon's genes. There is a negative correlation between the amount of tryptophan in the environment and the rate of tryptophan synthesis. So you have a lot of tryptophan in the environment, you're going to have low synthesis by the cell. And if you have a low amount in the environment, you're going to have a high amount of synthesis by the cell.